Alright, ladies and gentlemen, this is going to be the next video on cellular respiration and going through and trying to piece together everything in cellular respiration that we've learned thus far. Let's get started on this next video. Now, food is not going to be small enough to go through and fit into our mitochondria. We're going to go through and consume that food, we're going to put it into our stomach, and then we're going to go through and break it into smaller pieces. When the food is digested, we're turning it into that molecule glucose. That is when cellular respiration starts, when we've gone through and broken our food down into that glucose, and then send that glucose into the cell to eventually be used by the mitochondria. Now, food's going to provide a variety of other functions in our body, but for right now, we're learning about how we take that food and turn it into energy. So let's go through and look at this diagram here. We're going to look at the 3 steps, and we're going to start off with glycolysis. Now, the first part of going through and taking this food and turning it into energy is we're going to have broken it down into our glucose, and we start the chemical reaction in the cytoplasm with the initial investment of 2 ATP. Now we're going to go through and get a couple outputs here. What are those outputs going to be? Well, it's going to be 2 NADH and 4 ATP and 2 pyruvate. So remember, we invested those 2 ATP, so we say we have a net gain or a net output of 2 ATP when we go through glycolysis. Now when we go through and create these pyruvate, that's when we go through and send them into the mitochondria. We still have bonds that we can go through and break, and we can use those to generate the power and the molecules necessary for the Krebs cycle. So we're going to go through and take 2 pyruvate, and we're going to get a couple outputs when we shove them through the Krebs cycle. We're going to get 2 FADH2, we're going to get 8 NADH, and then lastly here we're going to get 4 ATP for our energy carrying molecules. However, we're also going to get this waste product. We're going to get carbon dioxide, which is going to be expelled from our bodies via our lungs into the atmosphere. Now, once we've gone through and established these 2 energy carrying molecules, this FADH2 and NADH, we send this all down to the electron transport chain. Our goal is to get that electron transport chain to function. So FADH2, 8 NADH from the Krebs, and 2 NADH from glycolysis are going to go through and allow the transport chain to function and create that concentration gradient, which allows ATP synthase to generate anywhere from 30 to 38 ATP. Depends on which textbook you go through and look at. But this is going through and piecing it all together. It is these 3 steps. So let's make sure we understand this in a little bit more detail. Inputs for glycolysis are glucose and 2 ATP. Our outputs are 2 NADH, 4 ATP, 2 pyruvate for a total of 2 net ATP. Remember, it's 2 net. This is going to make the energy for the ETC. It's sending that NADH down, and it's going to make some ATP and does not require oxygen. Now, the process here of glucose going into pyruvate is shown on the right. And remember, we're making the energy carrying molecules in the cytosol or cytoplasm to send the pyruvate into the Krebs cycle. Now, the Krebs cycle, the inputs are going to be 2 pyruvate, and we're going to get out 2 FADH2, 8 NADH, and 4 ATP. The goal is to again make the electron carriers for the ETC. However, this is going to require oxygen now. So oxygen is now required when we go through this Krebs cycle, and some of the outputs, the excess gas that we're going to go through and generate is going to be carbon dioxide. Now lastly here, we're trying to go through and again make that electron transport chain function. We need to understand why oxygen is important in this next step. Now the last step here is going to be the electron transport chain. The inputs are 2 FADH2, 8 NADH from the Krebs cycle, 2 from glycolysis, and our outputs are going to be ATP. Now this requires oxygen because remember, when we go through and have these electrons present, at the end of the electron transport chain, they're going to be toxic to proteins and DNA. We have to go through and attach them to something. So what do we go through and attach them to? We attach them to some oxygen, also to some hydrogen ions, and we're going to make stable water molecules. So oxygen is that final electron acceptor that makes those stable molecules and results in the generation of water. The end result of the production in the electron transport chain is anywhere from 30 to 38 ATP, which is originating from that glucose molecule. All right, so did you learn? Well, did you learn a couple of things here? Did you learn the inputs and outputs for each step of respiration? Did you learn and review what happens during respiration? And then lastly here, do you understand how we're piecing all three of these together? This will be the end of the video. I will see you all in class tomorrow.